Hello everyone. In continuation to the MOOC course of Basic Epidemiology, today we will be covering the Module 2. Module 2 will consist of descriptive studies. So what is descriptive epidemiology? It is the first phase of epidemiological investigation concerned with the observation of distribution of the disease or any health related events in human population, the identification of characteristics with which the disease or condition under study seems to be associated. The various steps involved in a descriptive study is defining the population to be studied, defining the disease under the study, describing the disease in terms of time, place, and person, measurement of disease, comparing with the known indices, and formulating an etiological hypothesis. Coming to defining the population which is to be studied. It is the first step to define the population base. The study population has to be defined in terms of total number of participants, composition within the population in terms of age, sex, occupation, culture, socioeconomic characteristics and other variables. The defined population can be the whole population, a representative sample, spatial groups such as school children, etc. Defining the disease under the study. The operational definition of a disease is, it is a definition with which the disease or condition can be identified and measured in the defined population with a degree of accuracy. In disease which do not have pathognomonic signs and symptoms, the epidemiologist will have to frame his own definition, aiming at the degree of accuracy. Coming to time distribution, three kinds of time trends or fluctuations in disease can occur. The first is short-term fluctuation, another is periodic fluctuation, and the third one is long-term fluctuation. Coming to short-term fluctuation, it is the best example is epidemic. Epidemic is defined as the occurrence in a community or region of cases often in other health-related events clearly in excess of normal expectancy. Then, what are the common cause? Coming to common cause epidemics, single exposure or point source epidemics. Another is continuous or multiple exposure epidemics. Then propagated epidemics, that is person to person or animal vector like arthropod or animal reservoir. The third one is slow or modern epidemics. Coming to periodic fluctuations, seasonal variations or trends, which are prominent features of infectious diseases. Example, measles, varicella shows peak incidence during the early spring season. Then GIT infections, that is gastrointestinal infections, have a seasonal rise during summer months. Then cyclic trends, disease which occur in cycles spreads over a period of days or weeks, even months and years. Examples, measles, which appear every 2 to 3 years before vaccines were invented of measles. Coming to long-term fluctuation or secular trends. Secular trend implies changes in the occurrence of the disease, that is progressive increase or decrease of disease over a long period of time. Definite movement in one direction. Example, heart disease, lung cancer, diabetes, which are progressive in nature. Then tuberculosis or polio, which are regressive in nature. Coming to place distribution, the geographic pattern of disease provide clues about the etiology of disease. The variations may be classified as international variation, national variation, rural and urban differences, and the local distribution. Coming to place distribution, the host factors which influence the disease pattern in an individual are age, gender, ethnic groups, occupation, marital status, behavior, and socioeconomic status. Now, what are the measurements of disease? The measurement of disease is done in terms of mortality and morbidity indicators. Morbidity can be expressed in terms of incidence and prevalence, which I had discussed in the previous module also. Incidence means the number of new cases, and prevalence means number of new as well as old cases at given point of time. The next step is comparing it with the known indices. The essence of an epidemiological study is to make comparisons and to ask questions. Comparisons are made between different populations and subgroups of the same population. By comparison, it is possible to reach conclusions regarding disease etiology and also to identify group or subgroups which are potentially at high risk for development of certain diseases. Now the last step is formulation of an hypothesis. What is an hypothesis? It is a supposition arrived at from the observation or reflection. It can be accepted or rejected using the techniques of analytical epidemiology, which will be covered in the later on modules. Now, a hypothesis should specify the population, it should specify the cause being considered, the expected outcome, the dose response relationship, and the time response relationship, which should be complete. Example, cigarette smoking causes lung cancer is an incomplete hypothesis, and an improved formulation can be the smoking of 30 to 40 cigarettes per day causes lung cancer in 10% of smokers after 20 years of exposure. The point is that it is covering the whole parameters which can be included in which can be reflected. Now, the last is the uses where this descriptive epidemiologies or studies are used. It provides data with regard to the types of disease, problems, and their magnitude in the community. It provides information on the etiology of disease and helps in the formulation of an etiological hypothesis. Provides data required for planning, organizing, and evaluating preventive and curative services. Leads to path for further research with regard as a particular disease problem. Basically, descriptive epidemiology is the first step in any kind of epidemiological studies. It further leads for analytical and experimental epidemiology. Thank you everyone for your patience listening. In the next module, we will be learning about the analytical studies, which will be case studies, case control studies, and cohort study designs. Thank you.